All right. Can everyone see my screen?
Yeah.
All right. Okay, so as Joshua said, I'll be presenting this week and we'll be talking about the cell cycle, which is an important topic in cell biology. So before we continue into like the more advanced information, I guess, we'll go briefly over some background information. So first of all, as many of you guys probably know, DNA is packaged into chromosomes, which are made out of chromatin. And chromatin essentially consists of both DNA and proteins. And two other important vocabulary words are gametes and somatic cells. Gametes are basically the reproductive cells found in eukaryotic organisms. So in other words, they are like sperms and eggs. And somatic cells are basically any eukaryotic body cells that are not gametes. And it's important to know that gametes have half as many chromosomes as somatic cells, but that's something we'll go deeper into later in the presentation. And on the right side, you can kind of see a figure that depicts a human karyotype, which shows like all of the chromosomes. Okay, now, I'll just briefly go over the purposes of somatic cell division. In prokaryotes and other unicellular organisms, cell division basically leads to reproduction since they only have one cell. But in multicellular or eukaryotes, cell division is used for growth and development from a fertilized egg, as well as cell renewal and repair. And on the right side, there's we use sea urchin embryos as an example of the growth and development aspect. So you start from one cell, but then through cell division, you can grow and develop into a multicellular organism. Okay, so these are the phases of cell cycle, and there's mainly an alternation between two separate phases. The first phase is interphase, which covers around 90% of the cell cycle. And it's basically where all the cell growth and replication kind of happen, or DNA replication happens. It can be further divided into three subphases, with G1 phase being the first one, and then S phase, and then G2 phase. Cell growth occurs in all three of these subphases, and chromosomes or DNA is duplicated during the S phase, but that eventually condenses into chromosomes. And after interphase, we have the M phase, which stands for mitotic phase. And this consists of both mitosis and cytokinesis. Okay, so this is a figure that depicts kind of what happens during DNA replication. So at the top, you see that we only have one chromosome. And following DNA replication during the S phase, it'll turn into two separate two chromosomes, but now we can refer to them as sister chromatids. And through mitosis, they will separate into two separate cells. Each of them have like a copy of the original chromosome. And in the middle, there's something called a centromere, which is kind of where like the two separate chromatids meet. Okay, now we'll discuss mitosis. So mitosis is conventionally divided into 5 phases as well. And the first one is prophase, which is followed by prometaphase, and then metaphase, and then anaphase, and telophase. Cytokinesis, which is cell division, it basically overlaps the latter phases and completes the cell division process. Okay, now we'll go into each of the 5 different phases of mitosis. The first phase, as I mentioned earlier, is prophase. And during this subphase, we will see that chromatin begins to condense into discrete and observable chromosomes. Also during this phase, we will have something called a mitotic spindle begin to form. And this is basically a structure made of microtubules that controls chromosome movement during mitosis. So this mitotic spindle will basically begin forming in prophase. and all of the assembly of all the microtubules involved in the spindle begin at something called a centrosome, which is like depicted in this drawing on top. And the centrosome replicates during interphase and eventually migrate to opposite sides of the cell during prophase. Next is something called prometaphase. And during this stage, we have kinetochores, which are protein complexes associated with the centromeres found on each duplicated chromosome. And this is basically where spindle microtubules will attach to the chromosomes. So after the microtubules attach to the kinetochores of a chromosome, they can begin to move the chromosome towards the middle of the cell. And also during this phase, the nuclear envelope will begin to kind of break up and disappear. Okay, next we'll cover metaphase and anaphase. After prometaphase, all of the chromosomes will kind of be moved to the center of the cell along an imaginary plane called the metaphase plate. And this is basically an imaginary structure, like it's not something that's really discrete, but it's somewhere in between the two poles of the spindle. And then after metaphase, we will see anaphase, in which sister chromatids of each duplicated pair of chromosome will separate and move towards opposite ends of the cell. And they do this by, of course, being attached to the spindle microtubules, which shorten at the kinetochore ends. So whenever they shorten, of course, they will pull the each chromatid to opposite ends of the cell. And finally, we have telophase. So in telophase, the genetically identical daughter nuclei will begin forming at each end of the cell. And the nuclear envelope will also begin to kind of pinch in. So this is kind of where cytokinesis is ready to kind of complete the process by creating the ultimate two daughter cells. Okay, so cytokinesis happens differently in plant cells and animal cells. So in animal cells, we will have a cleavage of the cell membrane, and this will form something that we call the cleavage furrow. On the other hand, in plant cells, we won't see the cleavage, it won't just be the cleavage furrow, and instead it's like a cell plate. So in this figure, you can kind of see the difference. So on the animal cell side, which is the left side, the cell membrane will basically just pinch in at the middle between the two nuclei, and it'll form two daughter cells. But in plant cells, you can see the vesicles start to form a cell plate. And when the cell plate is completely finished, it'll kind of form a new cell wall, and therefore completing the process of making two daughter cells. Okay, this is a unique form of cell division called binary fission. And binary fission is the form of cell division through which prokaryotes will reproduce. Essentially, the single chromosome in the prokaryotic cell will replicate at a point called the origin of replication. And daughter chromosomes will begin to move apart after it's replicated. And finally, when the two chromosomes are kind of separate, then the plasma membrane will pinch inward and form two separate cells. And since binary fission is like a process found in prokaryotes, it's generally believed that mitosis evolved from this process and opened the way for more complex and multicellular organisms to kind of duplicate their cells. Okay, so the eukaryotic cell cycle also requires regulation for it to function properly. And this is mainly due to the fact that cell division frequency will vary depending on the type of cell that it's occurring in. So it's generally found that cell cycle appears to be driven by chemical signals in the cytoplasm. And when this kind of moderation and regulation is disrupted, it can cause major flaws that can often lead to things like cancer. Okay, so this is an experiment that kind of led to the conclusion that molecules present in the cytoplasm are what control progression into S and M phases. So in the first experiment, we see one cell in the S phase and one cell in the G1 phase. And when you fuse the two cells, the cell in the G1 phase immediately entered the S phase, and its DNA began to like duplicate and synthesize more. But in the second experiment, they went one step further and combined an M phase cell and the G1 phase cell. And as they saw with the S phase experiment, the G1 cell also immediately entered mitosis, even without all of the like required steps before that. So like it didn't have like a replicated DNA. So, but it still regardless entered the M phase. So this kind of showed that it's not like the genetic material inside the nucleus that determined whether or not the cell was able to progress into the future, into like the next phases, but instead it was more of the cytoplasm, because when you merge the two cells, they obviously share a cytoplasm. So when this happened, the cell that was in the stage before the other cell, it also adopted the same stage. Okay, now we'll go further into the control system that kind of controls cell cycle. And it's basically a clock-like system with various checkpoints. So the three main checkpoints we'll be looking at will be the G1 checkpoint, the G2 checkpoint, and the M checkpoint. And the cell cycle will obviously stop at the checkpoints until the green light is kind of given. Okay, the G1 checkpoint is mostly regarded as the most important one in many cells. So once the green light is given at the G1 checkpoint, it generally means that the cell will continue the process and eventually complete cell cycle. And a red light here will cause the cell to exit the cycle and enter something that's called the G0 phase. Cells that are in the G0 phase will just continue performing their normal functions, but they will not enter like the cell cycle and try to divide. And so certain cells will actually enter the G0 phase indefinitely and like kind of stay there without ever reproducing. An example of this would be nerve cells. Okay, now we'll talk about two specific types of regulatory proteins that are involved in the cell cycle. So these regular proteins are called cyclins and CDKs, which stands for cyclin-dependent kinases. Essentially, the CDK, like as the name suggests, CDK activity is dependent on cyclins. So the fluctuating concentrations of cyclins kind of controls whether or not the CDK is active or not. And there's also a complex of where the cyclin and CDK is combined, and this forms something called MPF, which stands for maturation promoting factor. And you can kind of see in the figure on the right, on the bottom portion, it shows how the CDK and cyclin come together to form the MPF. And once this happens, it'll trigger the cell's passage beyond the G2 checkpoint and into mitosis. So checkpoints generally regulate cell cycle using both internal and external signals. And I'll give a couple examples of each. So an example of internal regulation is like an example, let's say kinetic cores are not attached to spindle microtubules by the time the cell reaches the M checkpoint. So if this is the case, then anaphase, like the cell will not progress into anaphase because, of course, the microtubules will be unable to move the chromosomes. And an example of an external signal would be growth factors, which are proteins that stimulate cell division. And there's also like other examples of external signals would be density-dependent inhibition and anchorage dependence. So density-dependent inhibition basically says that the cells will kind of control their cell division based on the density of the cells. So in the figure on the right, you kind of see that there's a plate of cells, and once it kind of occupies the whole surface area, the cells will stop producing or reproducing. And anchorage dependence says that the cells must be kind of attached to a surface, which is also why they will only reproduce when they are on the surface of the cell plate. And a lack of these sort of signals can cause cancer cells, which you can see happens in the figure as well. So as I said earlier, cancer cells can result when cells don't respond normally to the control mechanisms. And they may not even need growth factors for division because they have their own methods to like bypass this. So an example would be that the cells make their own growth factors, or they use something to convey the growth factor signal without actually having any growth factors present. And the process through which normal cells transform into cancerous cells is just referred to as transformation. And the cancer cells can then go on to make tumors, which are like large masses of the cancer cells. There are also two types of tumors that you should know, with one being a benign tumor, which is a tumor that does not reach the original site of the cancer cells. And there's also a malignant tumor, which is when the abnormal cancer cells will spread to surrounding tissues through like lymph vessels and blood vessels. So this causes metastasis, which is basically a process in which cancer cells form additional tumors elsewhere. In the figure on the top right, it basically illustrates the process in which a tumor becomes a metastatic tumor. So you can see portions of the abnormal cancer cells are able to spread to other regions of the body through the lymph vessels and the blood vessels. And this forms another tumors elsewhere. Okay, so that was basically a brief lecture on mitosis. And now we'll go into meiosis, which is a variation of the cell division by which gametes or reproductive cells are produced. Meiosis is divided into two separate sets of cell divisions. So the first one is Meiosis I, and the second one is Meiosis II. And by the end of the process, you'll have 4 non-identical daughter cells that each have only one set of chromosomes. So we call these cells haploid cells because, or it just refers to like having half of the number of chromosomes as the parent cell. So gametes, as I just said, gametes have half as many chromosomes as the parent cell, and we can call the parent cell diploid for having 2 times the number of chromosomes. Okay, so this figure kind of shows the whole process. So after meiosis, you can see that we have a sperm and an egg. Each of them is haploid. Let's say they have an n number of chromosomes. And upon fertilization of the egg, you will form a diploid zygote, which has 2 times the number of chromosomes because, of course, the sperm had half of it and the egg had half of it. And following mitosis and development, the one zygote cell can turn into a multicellular organism, which can then repeat the process through meiosis again. And this specific scenario is obviously human. So in humans, n would equal 23. So an egg and sperm will each have 23 chromosomes, and a diploid adult cell will have 46 chromosomes, which is 2n. So before we go into meiosis, I also want to point out or talk about the specific life cycle that's present in plants and some species of algae. So this is called alternation of generations, and it basically is a type of life cycle in which there's both haploid and diploid multicellular stages. The haploid generation is referred to as gametophyte, and these will reproduce and through gametes, and this happens through sexual reproduction. However, there's also a diploid generation that is called sporophyte, and they will produce spores through asexual reproduction. So after the spores are produced, they will multiply through mitosis and become gametophytes. So it's like it's a circular cycle. So let's say you start with the gametophytes, and then through sexual reproduction, you have two gametes that fuse together and form a zygote. And following mitosis, the zygote will produce a bunch of sporophytes, which then through meiosis produce different spores. So the whole cycle just repeats itself. So that's basically the alternation of generations. Okay, now we'll go back to the meiosis. So the first kind of step in meiosis is meiosis I. So this consists of prophase I, metaphase I, anaphase I, and telophase I, / cytokinesis, of course, which is when the cell properly divides. So I'll mainly focus on prophase I because that's when some of the important stuff happens. So in prophase I, pairs of homologous chromosomes, like after duplication, they will combine to form something called a tetrad. So on the top right figure, you kind of see a pair of homologous chromosomes, and together they form a tetrad. And also during prophase I, there is something called crossing over that occurs. And this is basically when non-sister chromatids will exchange DNA segments. So in the figure on the top right, you see that the blue chromosome and the red chromosome, they kind of cross over at some point. And then this results in the blue chromosome having a portion of the red chromosome and the red chromosome having a portion of the blue one. And the point at which the crossing over occurs is referred to as chiasma. So after prophase I, metaphase I, and anaphase I, and telophase I, they're basically the same as mitosis, except like instead of having one pair of sister chromatids, it's like a pair of homologous chromosomes. So in metaphase I, you will have a pair of homologous chromosomes lined up at the middle, and in anaphase, you'll pull apart the two homologous chromosomes. And through meiosis I, you'll essentially transition from a diploid cell into two haploid daughter cells. So after meiosis I, you'll have meiosis II. And this stage is actually very similar to mitosis. And once again, you'll just go from prophase II to metaphase II to anaphase II, and then finally telophase II and cytokinesis. So prophase I, that's a typo. That was supposed to be in the other slide. So on the figure on the right, you see that in prophase II, you have a couple chromosomes that have like the different recombinant chromatids that resulted from crossing over. And in metaphase II, they both line up at the metaphase plate, and in anaphase, they'll be pulled apart. This results in two or four cells actually, because you began with two daughter cells, and after division of each cell, then it'll result in four daughter cells. So it's very similar to mitosis in that aspect. Okay, so these are basically a summary of the three events that are unique to meiosis. So in prophase I, as I said earlier, there's crossing over that occurs between homologous chromosomes. And then in metaphase I, the tetrads, which are the pairs of homologous chromosomes, will line up at the metaphase plate instead of like an individual replicated chromosome. And this also means that during anaphase I, homologous chromosomes will separate instead of the sister chromatids. So in the figure, you see that you first see a figure of a cell in mitosis, and you can see that everything that lines up at the metaphase plate is just like a single replicated chromosome. Whereas in metaphase 1 of meiosis, you'll have like a tetrad, which is two homologous chromosomes. And finally, we will talk about genetic variation. Genetic variation is caused by mutations that arise during sexual reproduction. So basically, it'll occur as a result of meiosis. And the three main mechanisms of this genetic variation is 1, independent assortment of chromosomes, 2, crossing over, and 3, random fertilization. So I'll first go over random fertilization. So that basically refers to the chance of like a specific sperm for being the one to fertilize an egg. So there's so much sperm that is released, like the chance for a specific sperm to fertilize a specific egg is actually very low. So that already like causes some genetic variation. And 2, during prophase 1, you'll see crossing over between two non-sister chromatids. So this basically causes more genetic variation because, of course, they weren't from the same chromosome. And finally, independent assortment of chromosomes will also add to the genetic variation. Essentially, what independent assortment is referring to is that there are a lot of equally possible arrangements during metaphase 1. For example, you can see in the figure that during metaphase 1, you could either have the pink chromosome on the left or the pink chromosome on the right. And depending on this arrangement, you can have a set of 4 gametes that are completely different. So through outcome 1 on the left side, you see that the 4 gametes produced are like, there's one with 2 purple and one with 2 pink. But in outcome 2, that's not the case because of the independent assortment that occurs. So yeah, when you add up the 3 main mechanisms of genetic variation, it causes a lot of like overall variation in the gametes that are produced. And this causes each progressive generation to have a different kind of genetic variation. And that basically covers all of the presentation. And if you have any questions, you could either like say it out loud or type it in the chat. So if you guys can still ask some questions like if they come up, but if there's no questions right now, we can go into a Kahoot, which I'll put the link to in chat. My bad, it's a game pin, so I'll just share my screen. So here's the game pin. All right, I think that's so like some of the officers probably aren't joining, so I'll start the game now. Yeah, so most of you guys got this right. So interface is just, it consists of three subphases, which are G1 phase, S phase, and G2 phase. So M phase stands for the mitotic phase. Yeah, so most of you guys got this right again. And so the order in mitosis is basically prophase to prometaphase, and then metaphase, anaphase, and telophase. So I'm guessing the people who chose prometaphase likely just saw pro and chose it, but I don't know. So yeah, the first phase of mitosis is just prophase. Yep. So during prophase, like the spindle fiber, the mitotic spindle will begin to form at the centrosomes, but the centrosomes are not actually part of a chromosome, but they just form the microtubules that will eventually attach to the chromosome. So the actual part of the chromosome where the microtubules attach to is called the kinetochore. So basically, spindle microtubules actually shorten by depolymerizing at their kinetochore ends. So basically, where the microtubule is attached to the chromosome, and not the end that's attached to the centrosome. So I guess, yeah. So that's why the answer is actually false, because of the word centrosome. It should be replaced with like kinetochore. Yeah, so a cell that does not pass the G1 checkpoint will basically exit the cell cycle and enter the G0 phase, which is like a phase in which it doesn't divide. And I think I mentioned this during the presentation, but certain types of cells will enter the G0 phase and stay there like forever. An example of this would be nerve cells. Okay, so the answers are kind of spread out kind of evenly. So in a human diploid cell, you'll have a total of 46 chromosomes, which is generally referred to as like 23 pairs. But one pair of these chromosomes is the sex chromosomes, which is X and X or X and Y. And so if you take that out of the picture, like by autosomal chromosome, we refer to all the chromosomes that are not like the sex chromosome. So that would just leave us with 22 pairs. Yeah, so most of you guys got this right. And sporophytes are basically the diploid generation of like in an organism that goes through alternation of generations. And they will produce spores through asexual reproduction, which can then kind of divide into gametophytes, which is the other type of generation. So this is the last question. Yeah, so the blue answer choice would be correct if I said what is the end product of meiosis I, because at that point it will be two haploid daughter cells, but at the end product of meiosis as a whole, like you still have to remember there's meiosis II, and that will produce like kind of double the number of daughter cells again, so that'll become 4 haploid daughter cells. And the other two choices are incorrect because they say diploid, and during meiosis, only the parent cell actually has a diploid number of chromosomes. So yeah, that's the end of the Kahoot. Good job, guys. And yeah, that's the end of the presentation.